Okay friends, so now we are going to talk about the organization of the Golgi complex inside the cell. Now the presence of Golgi complex is really important for the protein sorting events and as well as the cellular trafficking events. The protein is made inside the endoplasmic reticulum lumen. Now that protein has to be carried out from ER via the whole system via this Golgi network towards the destination point. Now Golgi body is present between the destination point, which are organelles like mitochondria, plastid, lysosomes, and also the ER. So ER is the starting point, and all those plastids or mitochondria, for example, say the destination point. Now this Golgi apparatus is connecting these two things up here, and most of the time this Golgi apparatus is also helping the proteins to be secreted outside the cell or like into the cell environment. Now the secretory proteins are made inside the ER, and it is to be taken from the ER towards at the end of the cell membrane radius to just set those protein away from the cell, and that can be done using this Golgi apparatus. So now let us look at the organization of Golgi and how Golgi apparatus is maintaining its structure and how it is functioning. Okay, in endoplasmic reticulum or ER, the Golgi apparatus produces and process proteins. These proteins move from their site of production in the ER through the Golgi, after which they proceed to their destinations, either taking the residues residence in the plasma membrane, or being secreted outside the cell, or fusing with the endosome or lysosome. Okay. Now the proteins from the ER travel in transport vesicles to the ER Golgi intermediate compartments or ERGIC. Next they are transported to the region of Golgi called the cis Golgi network, where the proteins modified begins, and we can follow the maturation of this compartment by marking it with a dark color. We can see in the microscopic analysis, in the transmission electron microscopic analysis. Vesicle fuse with the cis Golgi network until it begins to mature and move into the Golgi stack, and then the ERGIC vesicle create a new compartments in its place. The compartments with their proteins mature to become the medial and the trans compartments of the Golgi stack, where the further protein modifications take place. So that is the vesicle scheme. Okay, so let me draw this scheme for you, because sometimes it becomes difficult to imagine all these things. So let me draw it. So here is the ER stack, and this ER stack, from the ER we are producing this vesicle. This vesicles is coming in to make this ER Golgi intermediate compartments or ERGIC, and in this ERGIC those small vesicles are bulged out and they come together to make the medial and medial cisternae, and this from this medial Golgi stack or medial cisternae some vesicles are again bulged out, and they come into this part of the Golgi, which is called the trans cisternae. So it's called the vesicle maturation model of describing the Golgi apparatus. That can be described in one way, and there is also another way of this Golgi apparatus, which is called the vesicular trafficking model. Okay, so this is the sorry, actually this is the vesicular trafficking model for describing this stack. Another model is there which is called cisternae maturation. We'll see later, but in this animation we are focusing on this vesicle translocation or vesicle translocation model. So vesicle is coming out from ER to make the ERGIC, and then again vesicle come out from ERGIC to make the cis Golgi, then again vesicle coming out from cis Golgi to make the medial, and from the medial it is try to make the trans. So that's how it's being done. The whole system is being carried out. Okay, so let me so that is the basic thing. So vesicle so the compartments of the Golgi gradually matures and progressively move through the Golgi in the cis to trans direction, carrying their components with them. So that's how the process is doing. Now the compartments, the trans Golgi network acts as a sorting and distribution center, which is directing the molecules to their final destinations like plasma membrane, secretion, endosomes, lysosome, and all this. The proteins live in vesicles, which bud off until the final Golgi compartment disappears and is replaced by a maturing compartment immediately behind it in the stack. So that is the case in this case. The transport vesicles that are associated with the Golgi apparatus functions to return the Golgi resident proteins back to the earlier Golgi compartments for the reuse purposes. The mechanism by which proteins move through the Golgi apparatus has been a long-standing area of controversy, but the model described here has the considerable support from the recent studies. That's why I called it, and this is called the vesicular trafficking model for describing the Golgi apparatus. Okay, there is also a cistern maturation model present. In the cistern maturation model, it has been suggested that the cis Golgi network is slightly sliding towards the medial Golgi region, and the medial Golgi region portion is sliding towards the trans Golgi region, and finally what present in the trans Golgi region is bulging out and go and fuse with the cell membrane. They start to make the cell membrane region. That's how the cis become medial, the medial become trans, and the trans become the part of the cell membrane. So this is called the cistern maturation model. This is not suggested here, and in this case what we are looking at, the vesicle trafficking. That means the vesicle is produced from the ER and fused with the cis, which is making the ER Golgi intermediate or ERGIC. From there we are making the cis, from cis we are making medial, and medial making trans. So that's how in this whole process is being carried out due to the presence of those vesicles and trafficking of those vesicles. That's why this model is called the vesicle trafficking model. Okay, and I hope that's going to help you understanding the Golgi trafficking. Thank you.